Hello and welcome to Biology Interests. Today we're going to be looking at meiosis, which is an important part in sexual reproduction, where you get two gametes, which are haploid cells producing half the normal number of chromosomes, joining together, fusing together to produce some kind of offspring. So what's important to know here is that in each cell you have 46 chromosomes essentially, which is the diploid number of chromosomes, 46 chromosomes each cell. What you need to know is that these cells, 23 of these chromosomes are from your mother and 23 are from your father, which is what we call a homologous, I'm trying to say it right, homologous chromosomes essentially. So you have 23 from your mother, 23 from your father, and in meiosis what they do is the homologous chromosomes, the one from your mother, one from your father, of the same chromosome, sit next to each other before the cell divides. So let's talk about this. But as long as you remember, homologous is meaning half your cells are from half your chromosomes are from your mother, half are from your father. It'll be good. So what we have to know about meiosis is that what you do in the first division, the homologous chromosomes almost pair up, and then you have their chromatids wrapping around each other, which we'll talk about crossing over in a minute in terms of cell variation. And then, so once that happens, the homologous pairs have separated, one from each pair going into each of the two daughter cells almost, okay? And then in the second stage of meiotic division, the chromatids then move apart, and by the end of meiosis 2, 4 of these cells have been formed. So we have 4 cells formed instead of the normal 2, which is what you get with mitosis. But each of these 4 cells only contains 23 of the normal chromosomes compared to the 46 you would get from normal meiotic cell division. Mitotic cell division, sorry. So let's just draw out a diagram to make it easier to understand about what's going on here. So we have the main cell here, the first cell. And now what this cell is going to have is, it's going to have 46 chromosomes, but I'm just going to draw 2. And so they're going to then line up in homologous pairs. So they're going to line up this one from the father and this one from the mother, and they're the same chromosomes. So they line up next to each other in the first step of meiotic cell division. One from the mother, one from the father. And then during mitosis 1, they get split into, all the homologous pairs get split and then randomly assorted, which is what another type of variation we'll talk about in a minute, randomly assorted into the 2 daughter cells. So let's say this one from the father or whatever gets put in that cell, and this one from the mother gets put in the other cell. And once that happens, mitosis 2 occurs, where they get split again into haploid cells. So these chromosomes get split into chromatids, meaning you have half the normal number of chromosomes. So one here, one there, one there, one there. So now we've got haploid cells, half the normal number of chromosomes. Okay, so that's basically how meiosis occurs. That's basically what happens. So you end up with one main cell, and then it basically splits into 4 haploid cells, which are sex cells, so egg cells, sperm cells. So now what can happen during this? What can happen during this is 2 main types of genetic variation. So one of these is called the independent segregation of homologous chromosomes. So if we write up here, independent, is that on there? So if we write up there independent segregation, and this is basically where, when you have the chromosomes lining up homologously, so mother and father next to each other, these can get split and mix up randomly, so the mother and father chromosomes can get split up randomly when they get placed into these cells. So these cells don't just contain all the mother's chromosomes and all the father's chromosomes when they split. They can have a random mixture of chromosomes from the mother, chromosomes from the father, and it's called independent because that's what happens. Each chromosome is separate almost. They can get randomly assorted into each cell randomly. There's no set order out of which ones have to go with which. So that's what happens there. So you get a random combination, and because they all contain different genes, different alleles, that means you get a different genetic combination in there. So that's a type of variety and type of genetic combination. So for example, let's say you had a cell here with chromosomes for, you had a chromosome for jumping, and you had one for blood group, almost. So let's just put this. Basically, then what happens is when the cell splits, you get a random combination of any of them essentially. So like you could end up with so basically with the independent assortment, this green chromosome here could end up with either the purple one or the red one when it gets randomly assorted. It's basically, it doesn't really matter. They're independent. They're not linked to one another. So as long as you remember that, it should be okay. And the other one is by crossing over of segments. And with the crossing over, what essentially happens is the chromosomes, basically the chromatids become twisted around one another. And during the twisting process, they almost break off. The bits almost break off of the chromosomes, and then they swap over essentially. So, and then that basically just means you've got different alleles on different chromosomes and a different mixture of different things. And so you could essentially have a cell here which might now have a red bit on it and a red one with a black bit on it, almost. And then they will go through the normal process of mitosis and end up like that. And that means you'll have a chromatid that is half a mixture of different alleles, essentially, and that introduces genetic variation that way. So I hope that's been fairly clear about the process of mitosis and how you go through the stages to get 4 gametes, and then you can also have variation by independent segregation, where each homologous chromosome is independent from the other homologous chromosomes and they can get randomly assorted, or crossing over where bits of the chromosome, when they're lined up homologously, can twist around each other, break off, swap over, and then that changes the alleles that way. Hope that's been clear. Please like and subscribe for more videos. Thank you.